of all. And it's good to see all of you today. Okay, so I would like to remind you that the submission of our report will be on Monday next week. Okay, so I think I have given you enough time to do your report for the first experiment. Okay, so last meeting we discussed some uh, computations about the pH of unbuffered solution and buffered solution. Okay, class? And we, there is a very important equation that we must remember always in order to compute for the pH of a buffer solution. And what do you call that equation? You may use our chat box. What do you call the equation that can be used to compute the buffer of the solution? Okay. Okay, the Henderson-Hasselbalch equation. Very good. Mr. Gamil. Miss Gamil. Sorry naman. Okay, so if we are to write the Henderson-Hasselbalch equation, it will be okay. So this is the equation for the Henderson-Hasselbalch equation. Okay, pH is equal to pKa plus log A minus all over HA. Okay. Okay, so, and we have some examples on how to compute for the pH of a buffer solution as given in our video presentation. So our topic for today class is about the cell. Okay, so wait lang. Class, pasensya na ha. Wala kasi akong headset dito eh. Okay, so for the first part, kindly compute class for the pH and pOH of the following solution. Okay, class. Let us first compute number 1. Find the pH, POH, and the hydroxide ion concentration and the hydronium ion concentration for HCl and okay, so let us do this first before we continue. Okay, as a recap of what you have understood about our topic last meeting. So I'm giving you 30 minutes to answer the question number 1 and number 2. Okay? And number 3 also. So you will upload your answer and I will present it later. Okay, for the first 30 minutes, please solve the following problem. Okay? I am giving you until 1:30 to compute for this problem. Sige, 1:45. Okay, please start computing for this problem. You have 30 minutes to solve all of this problem. This is an individual work. You have until 1:45 to solve for this one. This will merit 15 points. Okay, start answering now. Are you done? Class, are you still computing?
Yes po, we're still computing po.
Yes po.
Okay, sige, let's extend the time na lang and up to 2 p.m. Then at 2, we will start to discuss the different parts of the set. Okay? Okay, I'll just change the deadline. Bye! Oh, hintayin natin. Shh. Let's wait kasi bumili siya eh. Gusto niyo na ba umuwi? Siya, sige. If you like. It's okay kung gusto niyo na umuwi. Ako may klase ako, kaya mag-iintay. Sige, bye!
May pasa na naman sa'yo. Ay na lang natin, bitch. Oo, expect ko eh. Ayun na. Wala daw. Wala ba cheese dito? Eh, ito naman sa damutan siya, ito siya yun. Ang hirap kasi magmahal, tapos ikaw lang nagmamahal kasi. O, sir, may kinakay pa. May kinakay pa. Sa kaya kalisan mo. Naliit pala nung ang liit ng mga po. Kaya pinatarap ko din, natakamad ako din rin type eh. Yung sa Topaz, liit no, inaayos ko nga dito sa ginaya na kita, pinayaan ko sa tatlo. Kaya rin sila para yung sobra, pwede nila para tutubog kasi nga ang lutog nila. Ako sa yung iba yun, di ba? Mga kasi pa sila. Pero yung iba naman, isa, sobra ka lang ko. Pero yung mga talagang may sila Lala, sila Male, makaubos na mo. Kanina, na-review na namin yung 5, dito. Alam mo ba, may iba ka na ba? May iba ka na. Kasi nga, yung integers, yung integers, i-level namin. Tapos sabi ko, 60 items ito, kailangan lima-lima dosing araw yun. So, biglang mag-focus na. Alam mo yun, yung pinakatuwa mo, alam mo na ano mo din talaga. Kasi, parang sila dito na sila nagpapimbestiga yung pamilya ni Bea kung kasi si Bea may na kung ang lalaki, ang background ng lalaki. Alam mo bang bigla ng lalaki niya, bigay sa kanya ng isang key na relation siya sa religion. Yung control. Tapos may gasolinahan pa na ibang bigay ang nagbigay sa gasolinahan. Yun ang mga, di ba model siya? So yung mga ano-ano-ano mo, bigay-bigay sa kanya ng bigay na nakarelasyon. Hindi kami yung pag-ibig na relasyon na kaya hindi na sila akala ko dahil sa pula. Hindi. Dahil pa yan. Dahil pinimbestigahan. Kasi magkakaroon sila ng permahan. Di ba uso yun yun? Permahan na kung pera mo. O yung nagpa-imbestigahan. Yung pala yung imbestigahan. Kung mamalog ka sa ibang luto. Tapos sabi ko sa totoo ba ito? Kasi dito sa ako minsan naluto din yung mabibig. Totoo daw yun. Kaya yung pala yung imbestigahan. Kaya pala training siya ngayon.
Hindi na trending nga. Sana sila dalawa lang, malay mo sila. Ano?
Na hindi ko kasi hindi naman na siya. Pero ngayon, hindi na yata sila. Parang mas na. Mas na siya.
Ang dami ni Bea.
Oo. Pero kumahal din talaga yun. Ganda. Ang hirap naman magiging sa iyo. Ang hirap din. Pinaparawan ng puso mo.
Kahit kasi magkakaroon ng mga puso. Tapos pala tayo ng punta. Ay, ama.
Oo nga, no? Bakit na wala? Gusto ko yung sinabi ko, bakit pala tayo ng mga nagpupuya. Pero hirap sa nakarawan ng institution kasi talaga yun. Paano kung tingnan mo Tapos puti na lang. Maya, papahinga muna. Ito, baka kumain na. Kumain ka dyan, ma'am. Ma'am, meron yung sa balaba. Sino nagbigay yung bouquet niya? Naka-receive yung isang balaba mo. Si Christine yung Christine. Ang secretary, ay yung may box. Sino may box? Si Christine. Siya yung may bigit na bouquet. Ewan ko ba sino nagbigay mo. Sa parang ano, sino nagbigay ng klase niya? Parang siguro, kasi galing siya dito. Sa floor na. Ibig sabihin galing sila mag-ano yung parang. Ibig sabihin, sa edad nila ng ganyan. Pero parang serious sila sa mga ano. Laban ko. Diba? Kasi tayo noon, parang hindi tayo masyadong seryoso. Ngayon lang na-tune na, pero noon, parang hindi ganun ako eh. Pagka tayo lang hindi ganun.
Silipin ko muna yung mga bago na.
Okay, are you done? Class? Ma'am, convert ko lang mo sa PDF. Okay. Pa-open ulit ng submission. Thanks po. Okay, sige. Last na ito, then we'll discuss the result. I think most of you already submitted your... Wait lang. Wait lang, Miss Algisera. Ha? 16 pa rin yung aking submission. Oh, okay. I'll open this one, class. Please submit now and then we'll discuss. Don't worry. Okay, anak, inopen ko na uli. Sige, ayan, 26 submission. Okay, so let's start discussing how to compute for number 1. If ever you have questions, just raise your hand, okay? So, for number 1, sige, but before I start pala, okay na kayo? Nakasubmit na ba lahat? 29 tayo. So, ako na lang yung ako yung pang 27. May isa pang hindi nagsasubmit. Okay. Si Rain.
Sorry,
Rain?
ako po hindi pa po ka nakasubmit. Hindi ko po kasi ma-convert into PDF.
Ah, ano bang ginamit mo, anak? Ano?
In-scan ko po.
In-scan mo. PDF na yun. JPEG.
Ah,
Sige, palitan ko yung
try ko na lang po
submission.
ah, try ko na lang po i-upload ulit.
Wait lang ha. PDF, JPEG ang iyong ginamit, anak? O PDF?
Um,
PNG?
Kaya ko po ata i-upload as PDF. Baka ano lang po sa canvas ko.
Sige, anak. File upload. Hindi kita naman ako naka-receive. Media recordings. Sige na lang. Try ko rin yung media recording. Nag-try na rin ako ng text entry.
Ah,
Baka pwede ka na.
okay po. Wait, try ko po ulit.
Okay, we will discuss. Okay, so Rain already submitted her paper. Okay, so let's start. So again, let us look here. The equation for your uh, pH, if it is a buffered solution, is this equation, the Henderson-Hasselbalch equation. pH is equal to pKa plus log A minus divided by HA. Okay, now, if it is an unbuffered solution, our equation is simply pH is equal to negative log H3O plus. Okay, so allow me to share my screen. Okay, so for number 1 class, we are asked for the concentration, the pH of hydrochloric acid with a concentration of 0.001 molar. So using the equation, pH is equal to negative log H3O plus, the concentration will be, the pH will be 3. Did you get this one, class? Did you get this answer? Please raise your hand so that I can see. Okay. So, thank you. So, that is for the first problem. Okay. Now, since there is no POH, class, we don't have POH for the POH, of course, for hydrochloric acid. How are we going to get the POH for hydrochloric acid? We need to use the so there is no POH in hydrochloric acid because as you can see from the equation, there is no hydroxide ions. Is this clear, class? When hydrochloric acid dissociate, it will produce hydronium ion and chloride ion. There is no hydroxide, so we cannot write the POH for this one. Okay. Now, for the next one, we are asked about the points, the POH of this one, sodium hydroxide. So since the concentration of hydroxide ion is 0.001 molar based on the dissociation equation, based on this equation, If sodium hydroxide will completely dissociate, okay, so the hydroxide concentration that we will get will be 0.001 molar. And our POH is negative log OH, and that is also equivalent to 3. Okay ba to? Class, is this clear? Again, we, for the third one, for the second one, we are asked about the pH of a solution with the given Ka. If the Ka is 1.8 times 10 to the minus 5. Now, we all know that acetic acid will dissociate into hydronium ion and acetate ion. So, Ka is equal to hydronium ion multiplied by acetate ion divided by the quantity of your acetic acid. So, what will be, okay class, at equilibrium, the amount of acetate class is equal to the amount of hydronium ions. Okay? Kaya naging H3O plus squared itong ating equation. So, 1.8 times 10 to the minus 5 is the Ka of acetic acid. 0.01 is the concentration of our acetic acid. So, we will multiply Ka with the concentration of acetic acid. And we will get the following equation. 1.8 times 10 to the minus 5 times 1 times 10 to the minus 2. So combining similar terms, 10 to the minus 5 multiplied by 10 to the minus 2 is equal to 10 to the minus 7 because you need to add them. And 1.8 multiplied by 1 is 1.8 times 10 to the minus 5 equal to hydronium ion concentration squared. So, in order to get the value of the hydronium ion concentration, we need to get the square root of 1.8 multiplied by 10 to the minus 7. And what will be the equivalent? So we need to get the square root from here and then and this will be our concentration of our hydronium ion. Okay? So, paano ni ma'am nakuha yan? So, 1.8 times 10 to the minus 7 class is equivalent also to 18 ay, 18 times 10 to the minus, sorry class, 18 times 10 to the minus 8. Parehas lang yan class. This is not negative 3. It should be 4. Okay? So, 18. So, I have a calculator here. Share ko. Nahanap yung calculator ngayon. So if I write here the number 18, I'll write log and get the log of 18, and that is equivalent to, but naiba naman. Not yet. Square root pala to, class. Sorry. So I have here square root, and I'll write the number 18. So I get 4.24. Okay? That's why so I get 4.24. So the concentration of hydronium ion will be 4.24 times 10 to the minus 4. And then your pH will be approximately 3.373. Did you get that one? Class, did you get the value 3.373? Nakuha nyo ba yun, class? O iba yung sagot nyo sa akin? So, eto 4.24, di ba? Paano ni ma'am nakuha yon? So, I'll get the log of 4.24. So the log of 4.24 is 0.62736, and I have to subtract this one to 4, to the number 4. Okay? That's why I got the value of the pH as 3.373. Okay, class, did you get that one? Ayun, sige. Don't worry, I will give you a copy of the procedure on how to solve this problem. Okay, so that when you see this one in the exam, you already know how to calculate for the pH using Ka and the pH of strong acids and strong bases. Okay. Oops. Okay, so for the pH of the buffer, we will use the Henderson-Hasselbalch equation. Okay, and the pKa of our formic acid is 3.74. 3.74. Then we will get the log of the acid and the base divided by the base. So our log of the base divided by the acid. So wait lang. So 3.74 plus what? The negative log of 0.90. The negative log of 0.90 and okay. So we need to use ice here, class. Okay. For this one, we need to get the equilibrium concentration of our buffer solution. So we need to add 0.90 plus x, and also 0.90 x lang pala dun sa kabila. X is the amount of what? Of the base that will be produced from our acid. So we need to get that first.
2 hours mag-order sa kanila.
Okay, so ayun. So that's the how you compute for the pH of the buffer. Uulitin ko yan uli class in the classroom. Okay, so our next topic for today is a very easy. It's about the different parts of the cell. So, okay. We all know that the cell is the basic unit of life. So, today we will be looking into the different organelles that are present in the cell. So, first, we need to differentiate the different types of cells, distinguish the different organelles in the cell, and recognize the various biochemical system that exist in the cell, and correlate the composition and function of the organelles with the biochemical system associated with it. Okay. So there are two types of cell class. One is prokaryotic and the other one is eukaryotic. Okay. So, ako, and for our pre-task, we were asked to answer the question, how did life begin? So, sige, mag-relax muna kayo after nyo mag-compute. Sige, sagutin na nga rin natin tong how did life begin para naman ma-unwind ang mga isipan nyo. Can non-living organism produce living organism class? Is it possible for a non-living organism to produce another living organism? You may use the chat box. Pwede bang gamitin ang chat box? Pwede ba na mag-produce ang non-living things ng isang organismo? Pwede ba yon? Ay, napagod na ba kayo? Okay. So, okay. Is it possible that a living organism, a non-living organism or non-living thing developed into a living organism? Is it possible? No po. Okay? No po. Okay. So, hindi pwede na ang isang may buhay ay magmumula sa isang walang buhay. Palaging ang buhay ay nagmumula sa isang may buhay din. Okay? Kailangan natin yung tandaan. Okay, so, at dahil doon, ayan, meron siyang, wait lang anak. There is one comment. No, it's not possible for a to develop a living organism arise from pre-existing. Yes, you're very much correct, Eliza. Okay? Through process like reproduction, growth, and development. So, living organism has its own characteristics, very much different from non-living things. Okay? That's why one one cannot produce the other. Okay? Because life begets life, sabi nga nila. Okay.
Balae.
Sige, salamat balae ha. Okay. So, okay. So there are two types of cell, the prokaryotic cell and the eukaryotic cell. When we say prokaryotic cell, it means it does not have any nucleus. Eukaryotic cells have distinct nucleus. Okay, as you can see from the illustration here. Okay, so you will see that the DNA in a prokaryotic cell is scattered in the plasma, in the cytoplasm. However, for the eukaryotic cell, the DNA is located inside the nucleus. Okay, you cannot find the DNA outside of the nucleus in the eukaryotic cell. So these are the characteristics of a prokaryotic cell. They do not develop into multicellular form. So they are always unicellular and they are identical and capable of independent existence. They lack nucleus and membranous organelles. This includes bacteria and archaea or the archaebacteria. Eukaryotic cells, on the other hand, are made up of a nucleus and a membrane-bound compartments called organelles, which have specific metabolic activities. It includes fungi, animal, plants, as well as some unicellular organism. Okay, so the, the main difference basically for prokaryotic cell is the and the eukaryotic cell is the existence of a nucleus. Eukaryotic have true nucleus. Prokaryotic does not have a nucleus. Okay? So, these are the different parts of a eukaryotic cell. One, it has a cell membrane. So this cell membrane is a semi-permeable membrane, which means it selects the materials that gets in and out of the cell. Okay, so choosy. It this cell membrane is very choosy. Okay? Then we have the cytoplasm. The cytoplasm is where you can find your organelles. It is the living substance of the cell. While the cytosol, it is the fluid portion of the cells. Then we have the different membrane-bound organelles that can be found in our cell. First and foremost is our mitochondrion. The mitochondrion is the powerhouse of the cell. It is the one that produces our adenosine triphosphate. Cellular energy is produced in the mitochondria. Okay? Now, for the endoplasmic reticulum, this one is responsible for the intracellular transport. Okay? So, where is that endoplasmic reticulum in our cell? This one is the endoplasmic reticulum. This is how it looks like. Okay? There are so many folds in our endoplasmic reticulum. Then we also have the Golgi body. They are the packaging center of the cell. They transport the protein, okay, from the cell. Then we have lysosome. The lysosome is known as, sir, thank you. The suicide bag of the cell. Okay, it is similar to our digestive system. The peroxisome detoxifies the cell. Then, the third major part of a eukaryotic cell is the nucleus. It is a rounded structure at the center of the cell that controls the metabolic activities. It contains the DNA. Okay? So, wait lang. Ano. So, that three parts is the cell membrane. Okay? The cell membrane, the nucleus, and the cytoplasm. As we mentioned earlier, the cell membrane is semi-permeable, which means that it is very selective in all the in all the substances that passes through it. Now, aside from being a semi-permeable membrane, it is composed of a phospholipid bilayer with embedded proteins. Okay, so let us look into that phospholipid bilayer. So you can see that part of the cell membrane. Okay, you see this one, the circle, the circular structure. Okay. So that is the hydrophilic endpoint. Now, this gray extension glass is the hydrophobic extension. So the phospholipid bilayer is made up of a hydrophilic substrate and also a hydrophobic substrate. It is made up of those two. That's why it is very selective in allowing materials to enter the cell. Okay? So when we say hydrophilic, it means water loving. When we say hydrophobic, it is what? It is afraid of water. Okay, so the inside part is hydrophobic, while the outside part is hydrophilic for our cell membrane. Okay, now the cytoplasm class contains the different nutrients that the cell needs. It also helps break down waste product and move materials around the cell. It is the living substance of the cell where you can find the different organelles. The cytosol class contains water and free proteins and other substances. It is where a major part of the cellular metabolism takes place. It also contains the cytoskeleton. So this is also the part that divides during cell division. So the organelles. So a while ago, it is very and the clarity of the picture was very uh, poor in the first part. But here, you can see here the endoplasmic reticulum. And then there are two types of endoplasmic reticulum. The rough endoplasmic reticulum. Remember that the rough endoplasmic reticulum has ribosomes attached to it. Now, ribosomes are very important in the cell process because they are the protein factory of the cell. Okay, which means that proteins are made in the ribosomes. Okay, now, so and that ribosome class is can be found in the rough endoplasmic reticulum. That's why when the protein is made from the ribosome, the endoplasmic reticulum transport it to the other parts of the cell. Now, lysosome, as we mentioned, is the digestive system of the cell. Okay? So, lysosome may not be present in plant cell. Same with centriole. Okay. Then we have the mitochondria. As we all know, as we mentioned a while ago, mitochondria is the powerhouse of the cell because cellular energy in the form of adenosine triphosphate is manufactured in our mitochondria. Then we have the plasma membrane and the Golgi apparatus. The Golgi apparatus is the packaging center of the cell. Yan. Okay, so nag-discuss na kaagad ako. Ito pala ang kanya mga... Okay, so here is the endoplasmic reticulum concerned with intracellular transport. Okay. So the smooth endoplasmic reticulum has no attached ribosomes. It is the site for the synthesis of cholesterol and phospholipid and involves in the transport of fatty acid and other lipids. And the rough endoplasmic reticulum is granular with ribosomes. It receives proteins that are synthesized by ribosomes. Okay? It is more lengthy, mas mahaba, compared with your smooth endoplasmic reticulum. Okay, this is the mitochondria. The mitochondria is moving. It is also known as the powerhouse of the cell. Sorry, class lang ha. Because they release energy for the cell in the form of ATP. It undergoes self-replication in a manner similar to bacterial self-division. Okay? It may contain the DNA. Then we have the Golgi complex. So it is a stocked flattened membranes transport substance to and from the cell. It modifies, packages, and transport proteins and produces lysosome. Then lysosome, as I have said, it is the digestive structure of the cell. It contains powerful enzyme capable of digesting or breaking down all organic material. It is also known as the suicide bag of the cell. Okay? Because anything that goes into the lysosome is broken down to the different organic materials. Then we have the peroxisome. It breaks down hydrogen peroxide into water and oxygen. It is good for the detoxification of alcohol and other toxic compounds. It is formed by budding off the endoplasmic reticulum. Then we have the nucleus. The nucleus is a rounded structure at the center of the cell and frequently the largest structure in the cell. It contains the DNA. As we all know, the DNA is the genetic material responsible for passing one the trait of the parents to its offspring. DNA contains the code for those traits for one organism to be passed on for one organism to another. Okay, so let's continue. So after knowing the different parts of the cell, let us look into the plant cell and the animal cell. Okay. So what is the major difference between a plant cell and an animal cell? Uh-huh. But so let us watch these videos for additional information about the cell, the plant cell, and the animal cell.
Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
Okay, class. So you have seen how the cell looks like. Okay, that's the beauty of the video that we watch. We will be able to differentiate really the prokaryotic and the eukaryotic cell. You can visualize their structure, the organelles inside, as the function of the organelles and the structure in the cells are explained. Okay, so let us now look into the plant cell and the animal cell. Excuse me. Okay, take note of the different organelles that are present in the plant cell, which you cannot find in animal cell. Please uh, be aware also of the different structure that are present in the animal cell but cannot be found in a plant cell. Okay?
Animal and plant cells share many similarities, since they are both eukaryotes, which means that they have a membrane-bound nucleus. Plant cells are often larger than animal cells. Well, except for eggs, the largest single animal cell is an unfertilized ostrich egg, which weighs around 1.5 kilograms. So let's discuss similarities. Both animal and plant cells have membrane-bound organelles.
Wait lang.
They also both have
Wait lang class na ano ako.
Animal and plant cells share many similarities, since they are both eukaryotes, which means that they have a membrane-bound nucleus. Plant cells are often larger than animal cells. Well, except for eggs, the largest single animal cell is an unfertilized ostrich egg, which weighs around 1.5 kilograms. So let's discuss similarities. Both animal and plant cells have membrane-bound organelles. They also both have cytoplasm, a cell membrane, a nucleus, mitochondria, rough and smooth endoplasmic reticulum, a Golgi apparatus, and peroxisomes. The nucleus is where the genetic information of the cell is housed. The cytoplasm is not to be confused with the cytosol. The cytoplasm is the entirety of the cell contained by the cell membrane. The cytosol, on the other hand, is the part of the cytoplasm not taken up by organelles. The cell membrane is an important semi-permeable division between the cell and its surroundings. It allows the cell to control its own biochemistry by only letting certain substances pass through it. Mitochondria are the powerhouses of the cell. They are specialized to perform aerobic respiration, converting glucose to ATP, which is the energy currency of the cell that is used to perform functions necessary for life. Both rough and smooth endoplasmic reticulum help with the production and storage of proteins. The rough endoplasmic reticulum is covered in ribosomes, which translate RNA into protein. The smooth endoplasmic reticulum makes lipids. The Golgi apparatus modifies proteins. Peroxisomes metabolize waste. Now let's get to the differences between plant and animal cells. One major difference is that plant cells have a rigid cell wall around their plasma membrane. It is composed of cellulose, providing additional stability and protection. As a result, plant cells have fixed angular shapes, while animal cells are mostly round and irregular. Another important difference between animals and plants is how they obtain their energy. Plants are autotrophs, which means that they produce their own food. Meanwhile, animals are heterotrophs, meaning that they must ingest their food. Plants produce sugars through photosynthesis, and then break down the sugar to produce energy. Animals consume other organisms such as plants to obtain sugar, which they then also break down to produce energy. Plants are autotrophs thanks to special organelles called chloroplasts, which are full of chlorophyll, a green pigment that captures light energy to drive the reactions in photosynthesis. Photosynthesis is the process by which plants use the sun's energy to make sugar. Once the sugar is made, it gets broken down in mitochondria. Another important difference between plant and animal cells is in their vacuoles. Vacuoles in plant cells can occupy up to 90% of the cell's volume and have a single membrane. One of their roles is simply to fill up space, but they can also have digestive functions similar to lysosomes. Vacuoles have enzymes that perform many functions and can store nutrients or provide space to degrade waste substances. Animal cells can also have vacuoles, but they are much smaller than the large central vacuole in plant cells. There are multiple of them, and they don't take up 90% of the space. Plant and animal cells both also have cytoskeletons, which feature microtubules, intermediate filaments, and microfilaments. However, these structures are arranged differently in plant and animal cells. Centrioles are present in all animal cells, but are only present in lower plant forms such as Chlamydomonas. Centrioles are microtubule organizing centers, which are structures from which microtubules emerge. Plants don't have centrioles, but instead have many small nucleation sites. In plants, only reproductive cells, known as gametes, can have flagella, such as the sperm of bryophytes. Some animal cells also have flagella. In humans, sperm are the only cells with flagella. However, many animal cells have cilia, unlike most plant cells. For example, we have cilia in our respiratory system to help move debris and mucus, in the female reproductive system to help sperm move towards the egg, and elsewhere. Animal cells have clearly defined lysosomes. However, it's still debated whether some plant cells have them. Lysosomes are membrane-bound spherical vesicles containing hydrolytic enzymes that can break down biomolecules. They are involved in cell processes like secretion, plasma membrane repair, cell signaling, and energy metabolism. Plant cells also have plasmodesmata, which are channels that connect two plant cells. An analogous structure in animal cells is the gap junction, which connects the cytoplasm of two adjacent cells. If you enjoyed this video, like and subscribe. You can also support me by following the link to my Patreon. If you have any topics you'd like me to cover, please leave a comment.
Okay, so please write in our chat box what are the structure present in plants that cannot be found in animal cell. Okay? Structure lang muna tayo. Okay, write it down. What are the structures present in plant that are not present in animal cell? Now, magbibigay ako ng isang example. Do we have cell wall? Do animal cells have cell wall, class? Do we have cell walls? Okay. Yeah. Cell wall. Rain is mentioning cell wall. That's right. Chloroplast and a large central vacuole. Okay. So let's talk about those three structure first. What is the function of the cell wall? Class, why do what why do what is the function of the cell wall for the plants? For protection and support. Sige, that's good. Okay, tama naman yon. To protect the plants and to support the plants. Okay, so the cell wall gives the plant a rigid shape. Okay, so that rigidity is very important for the plant for protection. And you are right. Now, do you think of any other reason why the plant cell needs cell wall? Aside from that, why do you think the cell wall needs to be have for its tensile strength? Okay, so that is also for support. Okay. Now, remember that our cell wall, not the plant cell wall, also function for the transport of materials in a plant cell. Okay? It forms channel that are needed to move fluid for the plants. Okay, so that's another function of the cell wall. Now, let's talk about the chloroplast. Why? Thank you, Miss Marfa, Rain, and Miss Mangulabnan, and Miss Baduria for your answer. Okay, I haven't read pala Eliza's answer. Oh, okay, this is okay. So the water balance, that's what I am mentioning, is important for the plants. For growth, it also facilitates growth and storage of reserve materials essential for plant growth, development, and survival. So nakikita natin, class, that even at the cellular level, every part has a distinct role. Okay, we can appreciate the uniqueness of the and differences of the cells. Okay, now let's talk about the chloroplast. What is the role of the chloroplast for the plants? What is the role of the chloroplast for the plants? Okay, according to Terence, it is responsible for photosynthesis. Yes. The chloroplast contains the pigment chlorophyll that participates in photosynthesis. Okay. Energy excitation for the light reaction of photosynthesis is done with the help of our chlorophyll. Okay, so now during photosynthesis, the plant is making its own food. Okay, and that food is our glucose molecule. And at the same time, it is releasing oxygen. Without the chloroplast, there will be no organelles that would provide chlorophyll for our plants. So that chloroplast is very important. Okay, so the last one is the enlarged vacuole. We know that plants need an enlarged vacuole for the storage of water. So it is known as the leucoplast. Okay? So now, let's talk about the animals. So if the plants are rigid, are animal cells also rigid? Okay? So we can assume are animals also are animals. Yes, ma'am. Are animal cells rigid? Class, rigid din ba yung animal cell? Okay, do we have cell wall? Do we have cell wall? No, we don't have cell wall. Okay. Now, why do you think no animal cells are not rigid? You're right, anak. Eliza, very good. Why do you think animal cells should not be rigid? Why is it that the animal cells need not be rigid? Try to imagine our body. We have different types of cells. Diba? Let's talk about the muscle cell. Okay. Animal cells should not be rigid to allow for flexibility and adaptation, according to Eliza, on to the changing environment and physiological requirements, facilitating function such as cell movement, shape changes, and tissue flexibility. Well, that's very much good. Kasi po, kung medyo it will na into different, yes, very good, Andrea. It will not develop into different types of cells, tissues, and organs. Okay. Remember, in just the human body, we have different types of cell. For the blood, for example, we have the red blood cells, the white blood cells, the plasma. And those cells look very much different if we look into it in the microscope. Why? Because each type of cells have different function. Now, imagine if we have cell walls. If our cells have cell walls, it cannot easily move, diba? And there will be exchange of gases will be difficult in our body. Exchange of nutrients at the cellular level. Okay. Remember, class, what the plant needs is only carbon dioxide, water, and sunlight, and some other nutrients maybe. But for us living organism, there are a lot of movement that is taking place, that are moving inside and outside of the cell. That's why it needs it need not have a cell wall. Okay. So, um, what is present in animal cell that is not present in plant cell class? Okay. There is one organelle that is present in animal cell that is not present in plant cell. Okay. And that organelle class is the centrioles. Okay. So the centrioles class is important during cell division in animal cell. It helps, um, it forms spindle fiber. Bye-bye, sir. That separate the chromosomes during cell division. Okay? So, this, I think it is very important for the maintenance of our chromosomal number, the centrioles, especially for the for our sex cells and body cells. Okay? So, now that we are clear about the different types of cell, the eukaryotic and prokaryotic cell, and also the plant cell and animal cell and the organelles, let us move on to the next topic. Okay. So it is much, uh, we will focus now in the discussion of our cell membrane. Okay. So, wait lang anak. So, kanina nakita natin yung cell membrane. Ay, hindi na ano. Okay, so wait lang anak ha. Ayusin ko lang.
Baka natatagal ito yung mga buhok-buhok. Facial, ano nga to? Mask eh.
Wait lang. So, so the cell membrane is the membrane that delimits the cell from its surrounding, control what gets in and out of the cell, provides attachment for the skeleton of the cell, receive and sends out stimuli, provides binding site and receptor site for enzymes and other substances. It allows for cell-to-cell recognition and forms specialized junction with cell membrane of adjacent cell. So we will look into the cell membrane itself and its part and how those parts contribute to the different function of our cell membrane. Okay? So, so you can see from this illustration that the following are the role of the germ cell membrane for structural support, for recognition, for communication, and for transport. So it's not just a covering, but it is a complex membrane that performs so many functions. Okay. So, so now, so as I mentioned, we need to look into the structure of the cell membrane. So our cell membrane has a phosphate head. This one is the phosphate head. And this phosphate head is polar. When we say it is polar, it is partially charged. And also, when I say partially charged, it has a positive and a negative endpoint. Okay? So, it is also the hydrophilic part of the cell membrane. Okay, now, then we have this fatty acid tail. As what we learned in our organic chemistry, fatty acids are non-polar, which means it doesn't carry any charge at all. It is hydrophobic. It is it has a fear of water. It will not mix with water. And it is composed of saturated fatty acid, this one, and unsaturated fatty acids. That's why it is non-polar. So this is our cell membrane. Those are the components of our cell membrane. Now, you see, this is our phospholipid bilayer located on the peripheral side of our phospholipid bilayer are the hydrophilic endpoint, and inside is our hydrophobic endpoint. So, what is so interesting about this one is that it allows protein to move in and out of this membrane. Besides, this phospholipid bilayer class has also some protein that are attached to it. Now, these protein are known as transmembrane protein, integral protein, and also peripheral protein. Later on, we will see the importance of all of these protein. Now, what is being, before we go into that, let us look into the transport or the passage of material that happens in our uh, cell membrane. We have the active transport and the passive transport. Now, the active transport requires energy and goes against the concentration gradient, while the passive transport does not require energy and goes with the concentration gradient. When we say, let's talk about the passive. First, when we say it goes with the concentration gradient, it means that the material moves from the area of high concentration to the lower concentration. Well, that is the movement of any materials, whatever it is, you move from high to low. Siyempre, di ba, pagka high na, there's no more space. There is no more space. Therefore, the molecules will look for a way to go to a more spacious part. So that's why it during passive transport, it goes from an area of high concentration to low concentration. Para lang yung na halimbawa naglagay ka ng ink or colorings, 'di ba? Pamula do sa pinaka-concentrated na part, it will diffuse into the other parts which does not have any food coloring when we talk of that. Now, active transport, on the other hand, requires energy. Now, in this case, whether the concentration is not high and it the concentration does not matter in the movement of the material because there is an energy required to move the material. This is evident in the sodium potassium pump. Okay. So, this one is a passive transport. It goes from an area of high concentration to low concentration. Now, look here. This one is also facilitated diffusion. It goes from an area of high concentration to low concentration. Now, this facilitated diffusion will pass through our cell membrane with the aid of your protein. Okay, now look here. So this one is an active transport. I-enlarge ko lang. Okay. So this active transport happens in our electron transport chain in the mitochondria during the production of ATP. So you will see that the molecules here move. Again, we have a transport protein. But now, energy must be required. Okay? So, this ATP class need to
Sa kanya nung siya yung
need andyan pa ma'am.
Ay, ay, nandiyan pa siya. Nakita ko siya. Kausap ko siya kanina.
Okay, now this ATP will react together with the protein in order for the materials to move from the outside of the cell to the inside of the cell. Okay, remember that the energy molecule ATP is needed for this purple material to move from the outside to the inside of the cell. Okay. So look here. The cell opposes the concentration gradient. See, they are very few here, but still they pass through the cell, through the protein. Okay, and travel through an area where there are many of the purple substance. Later on, when we study photosynthesis, you will be able to see this active transport. Okay, now, diffusion class is an example of a transport system that does not require energy. Okay? So, what are the material that is diffusing in our body? Okay? Now, look. The materials that diffuses in our body are oxygen, carbon dioxide, and nutrients. Now, the process of moving these materials requires no energy. It just moves from an area of high concentration to an area of low concentration. Okay. Okay. Oxygen, carbon dioxide, water. It is non-polar, so diffuses very quickly. It easily passes to and fro our cells. Carbon dioxide is polar but very small, so it also diffuses quickly in our cell membrane. Water, on the other hand, is polar but is also very small, so it diffuses quickly in our cell membrane. Now, aside from diffusion class, there is what we call as osmosis. So, wait lang ha. What is osmosis? Osmosis is the diffusion of water across a membrane. Remember that class. Osmosis is the diffusion of water across a membrane. What happens to your hand when to your skin in general when you are swimming? Your skin gets dry, right? Why? Because the water from our skin Why does our skin gets dry when we swim, when we are in water? And what I mean, hindi dry, nangungulubot pala. Sorry, class. Sabi, baka isipin pa nila, hala, nag-swimming kami, ba't nga ba na dry? Of course, after washing, it gets dry because of the salt. But it is nangungulubot. Why is this so? Because the water in our skin is moving out of our body system. It moves from high water concentration to low water concentration. So another example of osmosis, class. So wait. So what we need to remember when we talk of osmosis. First, there is a semi-permeable membrane. Okay. And secondly, it is the water that is moving from area of high water concentration to low water concentration. Okay, as you can see from this one. The water here has there is more water on this part as compared to this part. So the water moves from this part to the other part. Okay. Now, aside from that, class, aside from that, so what are the examples of osmosis? So, ano ang gagawin mo pag ang gulay ay nalantaklas? When you have your... Oh, sige. Baka sabihin nyo, itatapon nyo. Have you tried submerging your not fresh gulay and fresh na yung medyo lantang gulay? Have you tried placing your not so fresh vegetable into water, submerging it in water? Once you submerge your for example, especially the green leafy vegetable, your kangkong, your talbos ng kamote, alugbati, if you notice that it is getting dry, try to sprinkle it with water. And afterwards, you will notice that again, it becomes sturdy. Okay? So, nagiging rigid na uli yung stem ng kangkong, pati yung leaves, medyo nabubuhay. Tapos, ganon din yung iba pa na green leafy vegetable. Bakit? It is because of osmosis. Siyempre, pag nalalanta na yung gulay, maunti na ang tubig. So, pag nag-spray ka ng tubig, na-absorb nito ang tubig kasi yung ini-spray mo na tubig ay mas concentrated kesa sa doon sa iyong sa iyong gulay. Okay? Ngayon, pag ikaw naman ay nag-swimming class, mapapansin mo namumula ang mata mo. Kasi yung tubig sa mata natin ay lumalabas kasi mas mataas yung water concentration sa loob ng mata natin, okay, kesa dun sa tubig sa labas. Kasi may salt yon, di ba? Kaya may tendency na lumabas yung tubig from our eyes, and our eyes get red, okay? So, yun yung sinasabi na movement from an area of greater concentration to an area of lesser concentration, the moving out of water. Now, do you know, class, that it is not good to drink distilled water? It is not good to drink distilled water. Wait lang ha, class. It is not good to drink distilled water because it may cause bloating in our cell. Okay. Kasi napaka-pure nung distilled water, wala yung salt. So it is an area of high water concentration. While our cells inside, there is salt in our cells. Okay. And then, since there's salt in our cells, it has less water concentration. So the water will move in through that, in, will move into the cell, and it will enlarge our cell. Okay, so this one shows the movement of water, high water concentration to low water concentration. Low solute when we have high water concentration, and this one that has low water concentration has high solute concentration. Okay, so we have different kinds of solution. Isotonic solution, hypotonic solution, and the hypertonic solution. Class, for a while, kindly analyze the illustration. I'll be back. Excuse me for a while.
Mama, wala na.
Sino nanalo? Sino nanalo? Ha?
Bakit mo? Kasi naganda sa sugar ako.
Alam ako sino siya.
Ito yung kanyang, nanalo ko siya, pero
Ayan para yun siya. Oo.
Daniel,
Kaya nga. Alam, ikaw.
ayun yung nanalo. Actually, ma'am, kung sa tindaw, mas aso nga ang mali niya ito kasi. Yan dapat nila natin. Di ko alam mo. Si Merlin. Yun nga rin yung inaano niya. Kahit ako magpasa, syempre, nag-gets ka naman yung point ni Sir. Syempre, judges kasi hindi, Sir. Sir, kinausap ko rin kasi si Sir. Sige naman yan. Sila lang mo din yung judges.
Kaya nakikinig ko, sabi ko baka natalo si Eunice. Ang ganda kasi, technically, maganda yan.
Ako man kakausap ko si Sir.
Bigyan mo na lang ng special award. Si Hane.
Hindi ma'am, si Sir, si Han.
Ah.
Ano ko siya, papayag din siya mo. Pero, kung susundin talaga namin,
Hindi, judge's decision. Ganun lang talaga, anak. Okay, class. Kasi kung hindi rigid po, okay, balik tayo, anak. Thank you, anak, ha. Pero, sa pagkulay pa lang.
wala naman.
Oo. Isotonic. Isotonic solution, when we talk of isotonic solution, class, it means it has the same concentration of solute as that of inside the cell. Okay, class. So, what are examples of isotonic solution? Remember, class, dati uso yung IMO. Do you remember IMO? Who among you remembers IMO? Those are eye droplets. Okay. Those eye droplets have the same solute concentration as that of our eyes. Okay. Once before, when our eyes get tired and it gets red, we sometimes put a drop of IMO in our eyes to refresh our eyes. Okay. And that IMO is an example of an isotonic solution. Actually, I was still thinking about other isotonic solution. Okay, yung kasi yung iniinom, yung pagka nagde-dehydrate ka, that is not a isotonic solution. I am still thinking, class. Maybe I'll remember another substance aside from IMO. Then we talk of hypotonic solution. This is a solution whose solute concentration, is lower than the solute concentration inside the cell. So you can see here, an example of this one is distilled water. The real distilled water. Okay? So, look here, there are more purple inside the cell compared with that of outside the cell. So this one is a hypotonic solution. Now, the tendency of water here is to get inside the cell. So in a hypotonic solution, the water enters the cell. So when the water enters the cell, it slightly expands itself. Nung bata kami, we always say, once it's a hypotonic solution, we say the cell gets bloated kasi it slightly expands because water here, which is higher concentration, will go here inside the cell because the water here has less concentration than that of the outside. Then when we talk of hypertonic solution, it means a solution which has a higher concentration than inside the cell. So from here, you can easily see there are so many purple dots. So those are the solute as compared with that of inside the cell. Now, this also means that there is less water outside compared to that of inside the cell. And so the tendency of water here is to get out from the cell to the outside of the cell. Therefore, it leads to um, possible shrinking of the cell. And then, if the cell is stays for a long time in this kind of environment, hypertonic, it may plasmolyze. Plasmolysis is known as the shrinking of the cell, which leads us to the next topic, cytolysis and plasmolysis. So, here, cells placed in distilled water. Now, what is distilled water again? Is it hypertonic or hypotonic? Of course, distilled water is a hypotonic solution because there is no solute in distilled water. So the tendency of water is to get inside our red blood cells. Of, of course, if it gets inside our red blood cells, the cell will expand, swell, and burst. And that will lead to cytolysis or cell death. Okay? Now, if you place these red blood cells in a concentrated salt solution, now, the cells has more water as compared with that of the environment. So the water now will get out of the cell. So the cell will shrink and shrivel and plasmolyze. So the shrinking of the cell is known as plasmolysis. Okay? So, after knowing osmosis, the different kind of solution, isotonic, hypotonic, hypertonic, and the movement of water in a hypotonic and hypertonic solution that leads to cytolysis and plasmolysis. We will now go to facilitated diffusion. Okay? Okay, remember class plasmolysis happens in a hypertonic solution. Cytolysis happens in a hypotonic solution. So what is facilitated diffusion? So facilitated diffusion does not require energy. Usually, there is a transport protein like this one that will move high to low concentration. Molecules will randomly move through the pores in channel protein. So they have a passage. Okay. The channel protein serves as a passage of our materials from high concentration to low concentration. So what are the examples of materials that are moving to and fro our cells by facilitated diffusion? So we have our glucose and amino acid moving from blood into a cell. Okay? So remember that one? Glucose and amino acid may move from one blood onto another cell through facilitated diffusion. Okay, so what are the different types of transport protein? We have a channel protein like this one, which are embedded in the cell membrane and have a pore for material to cross. So this one. And then we have the carrier protein. It can change shape to move material from one side of the membrane to the other. So carrier proteins is needed by our enzymes in our body. Okay, so the carrier protein, they carry, of course, and move the material from one side to another. Channel 14 have a pore or have a path where our materials can pass through from one side to the other side of the cell membrane. The next one is active transport. It requires energy or ATP. It moves from material from low to high concentration or against concentration gradient. So the example of which is sodium-potassium pump. So in the active transport, we have what we call as exocytosis. So what is exocytosis? Molecules are moved out here. You see here, it is moving out of the cell through a vesicle that fuse with the plasma membrane here. This is how many hormones are secreted and how nerve cells communicate with one another. Okay, class. So, in our nerves, we have between two nerve cells, class. For example, this is nerve A and this is nerve B. There is a gap in between these two nerve cells known as synapse. Now, in that synapse, the the message from this or yeah the electrochemical substance from this one will be released, and then it will what it will fuse with the plasma membrane of these next neurons. Okay, in or that will enable this cell to transmit the signal to the next cell despite that there is a gap in between. Okay, so this is the one that carries the for our nerve cell. This one is the one that carries the neurotransmitter. Then it will open up and release the neurotransmitter. Then this one will accept the neurotransmitter and then pass it on to the next nerve cell. It happens even if there is a gap between the two nerve cells. The last one is endocytosis. Now, endocytosis is the gulfing. Okay? So when we talk of endocytosis, large molecules move materials into the cell body by one of the three forms: pinocytosis, receptor-mediated endocytosis, or phagocytosis. So you see here that there is a bacteria from food particles. Now this is envelope. Okay? By the membrane. See that one? And then until the foreign body is encapsulated by into a vesicle, and then it is moved out of the cell. Okay, so pinocytosis and endocytosis. Okay, that is the last part of the lecture for our biochemistry. The succeeding days will will be experiment. So be ready for our experiment next meeting. Okay, the next experiment for next meeting is about biochemical processes. So before we leave, I would like you to know more about this that we will be doing in our laboratory. Okay. So, naku, meron pa palang metabolism at catabolism. So, we will be working on biochemical system by next week. Okay. So, we will be working on biochemical processes. Wait lang, anak. Hinahanap ko yung experiment. Transport processes. Dialysis. Ang hirap nitong ituro na walang... So, pag eto class, lecture na naman to. Alam ko meron tong ano. Yan, eto. We will be performing laboratory experiment 2 on Monday. Processes in a biochemical system. Dialysis. You will bring what? You will bring plastic bags for our dialysis, diffusion, hydrolysis. So you need to bring isa lang ha, wag madami. Isang elbow macaroni. Mali, sorry. 3 elbow macaroni. Okay? And a little of star margarine, which you need to apply in a slide. So, i-download ko na yan para i-send ko sa inyo. So, this will be our experiment for next meeting. It's a very easy experiment class. Okay? And then after that, after performing that experiment, wait lang anak. We will be doing the analysis for carbohydrates and the analysis for lipids. Okay, so those are, we need to finish that before the end of February. Okay. So, those are the activities in the lab. So, for the next weeks, we will be working in the lab and I'll see you on site. Okay? So, goodbye and thank you, class. Thank you for your time. And thank you for participating in our quiz. Please tell your classmates who were not able to attend our class today, okay, that we have a quiz. So that I can give them a makeup quiz, okay? Thank you so much, everyone. Goodbye. Thank you for your time.
Thank you po. Thank you po.
Thank
Ay, ma'am,
you.
ano po ulit yung dadalhin next experiment po?
Ano lang naman, margarine at saka ano anak, tsaka elbow macaroni. Anak, 3 piraso lang ng elbow macaroni ha. Anak, yung luto na. Okay? Okay ba anak? Yun lang.
Okay po,
And
thank you po.
plastic bag for the dialysis. Yung ano anak, yung food grade na plastic. Okay, pwede din ng cellophane pero mas preferred ko na yung food grade na plastic. Okay? So, I'll see you on Monday. Goodbye.